We're on the lymphatic and immune system. Put this in slideshow mode. This takes a little bit. Okay. Right. So the lymphatic system transports excess fluid away from the interstitial spaces in lymphatic vessels to prevent edema and to return leak plasma proteins and blood volume, actually, water back to the blood. So what are we saying there? When we looked at the activities that occur at a capillary bed, in the previous chapter for blood vessels, we talked about arterioles transporting blood into the capillary bed, indicated a diagram where it showed excuse me, fluid due to the hydrostatic pressure being pushed out of the capillaries into the interstitial spaces. And then because of the osmotic nature of blood on the venous side, it draws some of that fluid back in, but it doesn't draw all of it back in. So the excess fluid in the interstitial spaces needs to be taken up by the lymphatic system and transported back to the subclavian veins. Thereby, we are transporting excess fluid, the fluid that we lost from the cardiovascular system kind of temporarily into the interstitial space, back to the lymphatic system and back to blood, I guess I should say. Now, it lets you transport other molecules as well, but in doing so, we're kind of preserving blood volume and therefore blood pressure. It will transport fats in fluid into the bloodstream, when we look at the digestive system. And because of blood vessels transport lymphatic fluid, or I'm sorry, lymphatic vessels transport lymphatic fluid to lymph nodes. The lymph nodes will have different white blood cells there to monitor for the presence of microbes. So, when we look at the cardiovascular system here in the heart, we pump oxygenated blood from the right side of the heart. It goes out to the capillary bed, where due to hydrostatic pressure, a lot of fluid and different molecules and substances will be pushed out of the capillary bed into the interstitial space. But that a good amount of that will also be drawn back into the venous side of the capillary bed and return to the right atrium. But there's always fluid that, is then still remains in the interstitial spaces. So the lymphatic capillaries will end up taking up that fluid because there will be negative pressure inside these vessels for the same reasons that, or for the same reasons that, or not for the same reasons, but for the same dynamics that move venous blood back to the right atrium of the heart, valves, the respiratory mechanism, skeletal muscle contraction. We said that those are the ways in which venous blood will be returned to the right side of the heart. Well, the same occurs in the vessels, in these lymph vessels. And when you have compression on those lymph vessels and then it relaxes, it creates essentially a suctioning effect. That's what negative pressure is. So by doing so, it sucks up, kind of like a vacuum cleaner, fluid in the interstitial space into the lymph system. So then we can see here, fluid will be taken into the lymphatic capillaries, pass through the lymphatic vessels, from lymph node to lymph node to lymph node, until finally it returns to some of the subclavian veins and ultimately to systemic circulation. So lymph movement, as I didn't so articulately describe before, is that lymph movement is dependent upon muscle activity due to the respiratory movements of the diaphragm on the abdominal pelvic cavity, as well as the lungs expanding and affecting movement through the thoracic cavity, smooth muscle in the larger lymphatic vessels, and the valves that are present there. Let's remember, as skeletal muscles contract, it compresses the vessels that are passing through them. And so you compress it, because we have valves at different intervals. The blood maybe moves backward a little bit, but only as far as the last valve, because the valve is going to be closed. And the fluid has only one direction to go, and that's in a forward direction, opening up the next valve and passing on to the next vessel or right into another lymph node. All right. So when we look at the lymph nodes, we have vessels entering and vessels exiting. And once you know the direction of fluid flow and what direction, the lymph is traveling, the afferent vessel is the vessel carrying lymph into the lymph node, and the efferent vessel is the lymph that in the lymph vessel that's carrying lymph away from the lymph node. All right. So here, we have a picture again up on the top of the cardiovascular system, the arterioles leading to the capillary bed and then leaving in venous blood, and that how, get another schematic of how fluid is going to escape from the cardiovascular system. Normal thing to do. That which does not get drawn back into the venous side will be taken up into the lymphatic capillaries and then travel from lymph node to lymph node to lymph node. All right. The lymph entering one of those lymph nodes that are generally to the outer periphery, of that lymph node, they are the afferent vessels. When you have that one or two that's leaving the other side of the lymph node is called the efferent vessel. These lymph nodes are sort of shaped like a kidney bean, where the outer surface, the broader surface of that kidney bean will be the afferent vessels, and to the internal region and the indentation, the concave side of that kidney bean, would be where the efferent vessel would be found, transporting lymph to the next lymph node. Okay, lymph. We have lymph capillaries. Those are permeable vessels found everywhere but bones, teeth, and the central nervous system. We have lymphatic vessels that merge and form trunks. They collect lymph from the capillaries and transport it through the body. And then we have lymphatic ducts. So the ducts are the largest of these vessels or trunks. So, the pathway is kind of similar to capillaries, to venules, to veins, to larger veins, to an inferior vena cava or a superior vena cava. You're going to start with capillaries to vessels and trunks, and then ultimately ducts. Okay, so you have a right lymphatic duct and a left lymphatic duct. And the left lymphatic duct has always, and probably will continue to be, referred to as the thoracic duct. And I'll show you why in just a second. Okay, and when it comes to lymph transport, lymph from the right leg, the right arm, and the right side of the chest and the right side of the head do not all enter into the right subclavian vein. There's a funny different division, and it's not equal sides of the body are transported to a right and left thoracic duct, ultimately to the subclavian veins. There's kind of a weird division of it. So I'm going to show you in just a minute. So passing through the right lymphatic duct, which connects to the right subclavian vein, it will transport fluid from the right arm, the right side of the neck and the head, and to a certain point of the right part of the right chest. The rest of the body, the remainder of the chest, the abdominal pelvic cavity, right leg, left arm, left side of the head, left side of the neck, will pass through what's called the thoracic duct. The cisterna chyli is a dilated sac formed by the region of the merging of the intestinal and lumbar trunks, of the lymphatic system. It is dilated to accommodate more fluid. So let me show you this picture now. Okay, so here we have the gentleman stand here. The shaded area on the right side, excuse me, are the areas of lymph that will ultimately be transported to the right lymphatic duct. So you see here is right lymphatic duct. It connects to the right subclavian vein, just about where it merges with the jugular vein. So it's not the entire right side of the body that's going to, the fluid or the lymph fluid from the right side of the body that's going to pass through the right lymphatic duct. It's these discrete regions. And that's something really important to keep in mind clinically when these lymph nodes are enlarged or maybe hard. If they're spongy, it's usually not, it's more of a benign kind of an issue. But when they're enlarged and hard, like an uncooked pea of some kind, then you need to start looking at the areas where this lymph fluid is coming from that's passing through those lymph nodes, because that's generally not a really good sign, but it can be totally benign. So in this case, especially the right arm or the right armpit, the right supraclavicular lymph nodes here, you always want to look to the chest, to the arms as well, because any place distal, anything distal to that point could be the source of some kind of an infection or a cancer or something, that is now causing that lymph node or either to swell or has maybe metastasized to that lymph node. Okay. The rest of the body, now look at this: right leg, left, and the entire abdomen virtually, left chest, left arm, left subhead, and so forth. All that lymph fluid will pass through the thoracic duct. So here in the center, see where the inferior vena cava is here? And we have these lymphatic trunks merging here. We have them from the lower extremities and from the lumbar region, and they're all merging. It's like when 275 and 75 all merge together. You have like 2 3-lane highways, and the next thing you know, they're merging into one, and you're going to need 5 lanes before, after about a half an hour, mile or so, or a mile, you go from the 5 lanes down to 4 lanes and maybe back down to like a 3-lane highway. So when you have 2 large vessels merging together, you might have to allow a larger area to accommodate all the fluid that's passing through there. So you see that cisterna chyli? That's broader there and more dilated to allow all the lymphatic fluid being transported from the lower extremities and the lumbar area, enough room so it doesn't back up. Because remember, you're working against gravity as it is right now. You don't want to bring 2 3-lane highways together and make them narrow down to 2 lanes. Everything's going to back up behind you, so to speak. So you dilate that region so plenty of fluid can keep moving, and then it will narrow down a bit distal to it. Okay, so beyond the cisterna chyli, that tube system, that portion of that lymphatic vessel is named the thoracic duct because it's passing through the thoracic cavity. And it will ultimately circle around behind the jugular vein and articulate or connect to the left subclavian vein. So it's this real unequal distribution. Over here to the right-hand side, now you kind of get an appreciation of the critical nature and understanding that the right breast tissue and lung tissue will drain through ultimately the right lymphatic duct when you have nodal involvement either around the breast tissue itself, in the armpit, the axillary area, or what we call this chain above the clavicle called the supraclavicular lymph nodes there. Then you need to definitely look to the chest, look to the arms as well. Anything on the distal, on the other side of those lymph nodes, could be the etiology of these involved lymph nodes, either swelling or becoming larger and dense. We have primary lymphatic organs and secondary lymphatic organs. The primary lymphatic organs are where the blood cells that function in specific immunity and immunocompetence, it's called, come from. So we know that all blood cells come from red bone marrow. And that is where your erythrocytes, all your white blood cells, and your platelets are going to originate from. So the red bone marrow of spongy bone is going to be one of those primary sites. And so is the thymus gland that's in the chest. The thymus gland produces a hormone called thymosin. And thymosin is a hormone that stimulates the maturity of what's called the T lymphocytes. That's called becoming immunocompetent, to be able to produce an immune response. And in the red bone marrow, our B lymphocytes will become immunocompetent or sensitized at that point. All right, so the primary lymphatic organs are red bone marrow and the thymus gland, and the secondary lymphatic organs are essentially everything else, all the other structures, nodes, the spleen that also filters blood, kind of like a lymph node, but filters blood. All the other remaining organs, they're part of the secondary lymph, system. The spleen remember we looked at in the blood vessels, we said that we have um, as a aorta passes through the diaphragm and enters in the abdominal pelvic cavity, one of the first branches that comes out kind of right anteriorly, from the aorta is that celiac trunk. And one of the branches off that ultimately supplies the spleen. So oxygenated blood is going to go to the spleen. The spleen is going to act kind of like a lymph node, where you have all these reticular fibers there as part of the reticuloendothelial system. And it acts to capture, before we were talking about the spleen maybe capturing worn out red blood cells and breaking them down and producing the unconjugated bilirubin. Well, the spleen also monitors the contents of the blood entering it. It's going to spread out kind of like fluid as it spreads out through a sponge, and you have all the different walls of the sponge, and we will have white blood cells occupying those areas and monitoring for the presence of different microbes. So the spleen filters blood. The lymph nodes will do this similarly. It's going to spread out the lymph fluid that's being passed through those lymph vessels, and again monitor for infection. So lymph nodes filter lymph fluid. The spleen filters blood. Okay, so the red bone marrow is the production site of all blood cells, as I said before, including lymphocytes. Lymphocytes that become immunocompetent here are called the B lymphocytes. The thymus gland, found anterior and superior to the heart in the thoracic cavity, secretes thymosin. That is a hormone. That hormone stimulates T lymphocyte maturation or immunocompetence. That's the word they'd like to use, immunocompetence. Right. The spleen filters blood. So here's a picture of the spleen found in the left upper quadrant. We have an arterial supply goes in, and then the vein leaves. So when the artery enters, it's going to branch off, and you can see all those little branches. And then further, just below that, it gives you, like a diagram of what it looks like at the capillary level. All arteries, that supply those muscular arteries that transport blood into organs will branch off to a smaller, and ultimately the smallest capillary bed. It happens in every single organ. We need to narrow down those blood vessels to a capillary level, to that squamous cell, simple squamous epithelial tissue layer, for blood to, or microbes or nutritional products or waste products to move from the blood out to the tissues in those cells, and from the cells back into the blood. The same organization is going to be in every single organ. You come in as a large vessel, and ultimately everything has to narrow down to a capillary like that. And that's where all the action takes place. Okay, so the spleen is the largest lymphatic organ of the body. Good to know. It has sinus areas filled with blood, and it contains two types of tissues, white pulp and red pulp. The white pulp area is generally where you find mostly lymphocytes, and the red pulp is where we essentially find everything else. Mostly red blood cells, that's it's red, some lymphocytes, and some macrophages. So the white blood cells that we find, not only in the spleen, but in lymph nodes, are lymphocytes and monocytes or macrophages. If you ever get a question, and I've seen many NCLEX questions, and I never saw this asked, is if there's ever a question about which organ contains white pulp or red pulp, it's only the spleen. Okay, lymph nodes are clustered in different areas of the body, in the inguinal region, the axillary region, the cervical region. And the cervical region, arguably, depending on the textbook, because sometimes they group regions together around the head neck area, are 9 or maybe even 11, considered 11 different clusters of lymph nodes that are part of your head and neck assessment, when you're looking at patients and learning how to do this. So you evaluate, do good palpation using your hands to feel these different areas where clusters of lymph nodes, are located in the cervical region. It's common, or well, it may be, it's common to palpate the axillary region, because those axillary lymph nodes would be change in their size and palpatory characteristics when you have issues either in the arm distal to it, maybe in the breast or chest area of that same side. When it comes to the left side, the lymph that's traveling through that thoracic duct, to the left subclavian vein, that lymph is coming from all over. And remember, that's transporting lymph from the right leg, the left leg, the abdomen, left arm, left side of the head, and so forth. So when you have nodal involvement, in let's say the inguinal area, which is the bend at your hip there, by the pubic area almost. It's like, okay, well, if it's on the right side, then we have to examine and look at structures or a source distal to that point, in the leg somewhere, maybe in the thigh or the lower in the foot. And in the left side, it would be the same. If I had nodal involvement in the inguinal area of the left leg, then I would look to the thigh and the lower extremity. They function and they act as lymph filters. Essentially, lymph in those afferent lymph vessels are going to enter the lymph node. It's going to allow that fluid to spread out and pass through all these chambers within the lymph node, kind of like taking a sponge and putting it in a little bit of water. The water kind of gets sucked up into the different chambers of the sponge, and then you can wring out the sponge and the fluid will be, ejected away from the sponge. Well, lymph nodes are the same way. We're going to bring fluid to that more convex surface of the lymph node, and we're going to allow that fluid to get spread out and pass through all the different regions and little chambers of that lymph node. And the walls of those chambers are going to be lined with these T cells, B cells, and macrophages, the lymphocytes and the macrophages. And that allows that fluid to be thinned out nice and thin so that no one can hide inside there. That fluid is so thin that the cells lining the walls of those different chambers can identify microorganisms if they are present. Once that's done, it recollects on more of the concave side of that lymph node, and it'll exit as an efferent vessel and possibly go to another lymph node and another lymph node, all the way back to the respective right and left subclavian veins. So these lymph nodes, are areas where we can, monitor for the presence of different microorganisms and then stimulate an immune response. Okay, they're classified as an organ and has connective tissue capsule to the outside of it, those lymph nodes. It has a cortex region, which is always the outer region, that contains B cells, T cells, and macrophages. And the medullary area that also contains B cells, macrophages, and plasma cells. And remember, the plasma cells are a B lymphocyte that are producing antibodies. So on the most part, the cells to the cortex are pretty much the same cells in the medulla. There's slightly different, there's a difference between how many more B cells are in the cortex versus the medulla, and macrophages in the medulla more than in the cortex. It's not of real consequence. There are somewhat of different percentages of populations in the cortex versus medulla, but pretty much the same cells can be expected to be found there. B lymphocytes, T lymphocytes, macrophages, and in the medulla we have more B cells, not as many T cells really, macrophages, and the plasma cells are the B lymphocytes that are producing antibodies. Then you have the sinus region in a lymph node that collects lymph and sends it to the lymphatic vessel. Okay, in the digestive system, and we're going to come back to this, when we get to the digestive system, this model to the right represents a sliver of the tissues that we would find in a small intestine. And we have those little undulating waves up at the top. Those are called the villi, of the mucosa, of the tissue that forms the intestine. The intestine is composed of, multiple layers. You have the mucosal layer, and we'll see these same layers come to repeat when we look at the respiratory system. You have a mucosal layer, a submucosa. We have a muscular layer here composed of 2 layers of muscle coursing in a different direction, down here at number 1 and number 2, and there's a C over there as well. Right, so these little finger-like extensions up here, are extensions of what's called the mucosa. It's composed of simple columnar epithelium. And in each little extension, is a capillary bed in red and blue here, and then a green tube system, D. Right. That is a lymphatic capillary. So these little finger-like extensions extend into the lumen of the small intestine, and it increases the surface area for absorption of molecules and fluids and things that are passing through your small intestine. So, if you could see your small intestine, and you look at the layer that's exposed to the food that's passing through your small intestine, you have these like little finger-like extensions into the lumen. It's sort of my best analogy is hair bristles. And you have a brush and all the different bristles extend. If you have these little bristles extending from 360 around the wall of that into the center of the small intestine, and you could absorb molecules of across the surface of each bristle of this brush or comb, then it would increase the surface area. You would be able to absorb a lot of molecules. Well, that's what's happening here in the small intestine. Okay, so in each finger-like bristle or extension is a capillary, a lymphatic capillary, and it is able to absorb molecules that are being absorbed across the surface of this villi, and transport it. That's just the bottom line. We'll come back to this when we look at digestion, but you have to understand that on the surface of these little extensions here is that there's food passing, molecules you're breaking down in digestion in your small intestine, and those molecules can be absorbed. The fats will enter into the lymphatic vessel here, D. At the base of it, you see that blue ball, the blue ball there? That's called a Peyer's patch. Okay, so location, aggregations of lymphoid tissue that are usually found in the lowest portion of the small intestine, the ileum. These are the Peyer's patches. They function in surveillance of the intestinal lumen and in facilitating the generation of the immune response within the mucosa. So we have a lymphatic vessel here. They transport the fluids that they absorb, and it passes through and around this Peyer's patch. And the Peyer's patch, again, is going to have T lymphocytes, B lymphocytes, and macrophages. And so as material in the gut passes into our body, it can be analyzed as well as far as the presence of different microbes that might be, have some, in the food that you've eaten, and it's passing through your small intestine, and now it's getting absorbed into your body. Okay, so those are the Peyer's patches. That's the blue part. The lymphatic capillary is D. Okay. Lymph nodules, also called the mucosa-associated lymphatic tissue because of their locations, right? So these lymph nodules, examples, are going to be, or this molt tissue is going to be your tonsils, those Peyer's patches we just looked at, and your appendix. We haven't looked at that yet. Okay, so, we just said before, we have lymphatic capillaries. They merge into lymphatic vessels and trunks and blah, blah, blah. They pass through lymph node to lymph node to lymph node to the subclavian veins. If they're not technically lymph nodes, we do have other structures. All right, that's why they call it mucosa-associated lymphatic tissue. It's not technically a lymph node, but they function to help monitor, the contents of, let's say, in the case of the Peyer's patches, organisms that might be getting across the small intestine, in the tonsils and the adenoids, which are located in the upper part of your throat, in different areas of your throat called the pharynx. They're monitoring the, microorganisms or something that, we need to be aware of, like allergens that you're either breathing in through your nose that's hitting the back of your throat, or maybe the food that you're chewing and swallowing in your mouth. Either way, from either source, they're going to pass these tonsils or this lymphatic tissue, specific lymphatic tissue found there. All of these, as well as the appendix, which is kind of an obsolete organ, at this point. The appendix is a little remnant, of your immune system that looks like a little tail at the base of what's called the cecum, where the small intestine meets the large intestine. You have the ileocecal valve, and just distal to that point, there's a little sac inferior to that point called the cecum, and at the most inferior aspect of that is this little tail-looking structure, and that's called the appendix. It's thought that played an integral part in stimulating your autoimmune disorder, but at this point, it's kind of considered vestigial and that it doesn't really do a whole lot anymore. So all these areas, either the tonsils that are found in the pharynx, which is upper part of the throat, at the base of around the tongue, the Peyer's patches found in the ileum, the appendix found between the small intestine and large intestine, they help monitor for infection. They help remove pathogens from the area, kind of capture it, and then stimulate your immune system. Okay, so immunity, we keep saying immunity. What does that mean? It's our ability to defend really against infection from different sources, viral, parasitic, and bacterial. So when it comes to immunity, we have what's called innate immunity, which is I still refer to as non-specific because I think it's easier to, connect to that. And then we have adaptive immunity, which is our specific immunity. And when it comes to microbiology, should you take that next, they generally refer to it as non-specific innate immunity versus adaptive immunity. But innate immunity are the mechanisms in place to help generate a response to a pathogen or a foreign substance that is, the response doesn't depend on whether it's this organism or this one specifically. It's more general. These systems are initiated whether you get exposed to a bacteria, a virus, or you twist your ankle even, because that's going to be part of the inflammatory process. The same reactions are going to occur, or the same physiological responses are going to occur. Protection functions in the same way to each invader and confined to an infectious site or to a localized site. So I'm going to give you an example of that, in just a minute. Most mechanisms or physiological responses that we're going to talk about are going to fall into this innate, non-specific way of, responding. The specific or adaptive immune system is going to be organism specific, in that I have to be exposed to this particular virus. And then once that's, I'm exposed, we talked briefly about how, you have the A lymphocytes and the B lymphocytes. Then the B lymphocytes will make antibodies to the specific antigen on that particular virus. And the T lymphocytes, or a certain number of T lymphocytes, will start making granules and design how to kill that particular virus that has that particular antigen on it. So B lymphocytes and T lymphocytes can be cloned, and you can increase the number of them. But once they're sensitized, that one B lymphocyte only makes antibodies to one antigen. It doesn't do double duty. And the same with the T lymphocytes. It's going to make granules and design a method to kill the one organism that has a particular antigen that it's looking for. Now, you might have, 50 different B lymphocytes and T lymphocytes making, in the case of B lymphocytes, antibodies to 5 different organisms, 5 different viruses, let's say. That's cool. But no one B lymphocyte, one of those plasma cells, is going to make 2 different types of antibodies. They make them one class of antibodies, or one type, for one specific target that has that specific antigen on it. Right? So it's very specific. It's not going to be looking for E. coli or someone else. No, it just wants that one organism. And that's why it's so specific and why it works so well. All right, so the specific immunity is antigen specific. It recognizes a particular antigen as a specific invader. At the end of the process of all those B lymphocytes and T lymphocytes, you're going to have some leftover cells, I call them, and those are the memory cells. They hang out after you've conquered that virus, otherwise you'd be dead. And they linger so that if you ever get exposed to that virus again, you already have all the tools to kill it. You don't have to figure out how to make the antibody to fit on its antigen circuit like a lock and key. You already have the recipe. And when it comes to the T lymphocytes, you already have the recipe to make the granules for that specific, antigen in that organism. So your ability to respond and generate a massive number of antibodies in these granules, is accelerating. It takes a fraction of the time compared to your first exposure to a virus that you've never encountered before. That takes time to figure out and, how to, kill it and then clone all these cells and make all these antibodies and flood your plasma with them. So that's what we call having memory. And the adaptive, side of your immunity is systemic because we're going to use antibodies. We put it in plasma, and your T lymphocytes are found in, blood as well. As like, look, blood goes everywhere. So it's not contained into one confined infection site like we do for innate immunity. These cells and these antibodies travel all over. So it gives you more global protection. Okay, antigens. We talked about antigens in the blood chapter. Antigens are these large complex molecules, usually proteins. They might have some carbohydrate in them, some monomers of sugars in there. And they're found on the surface of cells. The ones that are on all our, nucleated body cells are those MHC complexes mentioned below. We have MHC1 complexes and we have MHC2s. Right. So the term antigen just means that it's some kind of large molecular, complex on the surface of the cell, which is an serves as an identifier. Now, antigens, if they're not consistent with my MHC complexes, are then deemed as a foreign cell. And so that antigen on a foreign substance or a cell can stimulate an antibody, production, an immune response somehow. All right. So reactivity, an antigen, ability of the antigen to react specifically with an antibody. If it's a self-cell marker like a major histocompatibility, then it won't initiate an immune response. If it's an abnormal one, or one that shouldn't be found there, is not consistent with my MHC complexes on all my nucleated cells, then I'm going to make antibodies. If it's the same, then I don't make antibodies to it. So our ability, the immune system, is based on the fact that we can recognize cells that should be there normally versus other antigens from foreign invaders. If you can't tell the difference, then you're going to have autoimmune disorders. So antigens on plasma membrane of nucleated cells, ours, are called major histocompatibility complexes. We have different types, MHC1s and MHC2s, but these are the MHC1s, basis for tissue typing for transplants as well. Antigens on RBC membranes is the basis for blood typing. Remember, RBCs are anucleated. So that's why we have all those. If you're A, you make anti-Bs. If you're blood type B, you make anti-As, and so forth and so on. Okay, so antigens may or may not be able to initiate an immune response. It depends on whether they're supposed to be there in their normal cells or found on our normal body cells, or if they're antigens found on invading cells or viruses. Right, so our innate responses are the ones, these are the non-specific responses. They are defense mechanisms on the surface of the body, like your skin, mucous membranes, secreting oil on your skin. Okay, the stratified squamous nature of your epidermis, that's a real physical barrier. Right, those things are there no matter what bacteria or virus is outside of your body. They still function. They're not specifically guarding against one versus the other. They're there all the time. Internal defenses would be the phagocytes like neutrophils and monocytes. Natural killers are a certain type of lymphocyte. We'll talk about that briefly in just a minute. Inflammation, antimicrobial proteins that come from those white cells, and fever, elevated body temperature. Those neutrophils and the monocytes, remember we said that they are, they phagocytize material, and they generally don't care. I mean, the neutrophils are pretty much bacteria only, but the monocytes, macrophages, they're like the garbage collectors. They don't care if you're a a cocci or a bacillus, or whatever, or a virus. It will phagocytize it. Right? That's what we mean by like non-specific innate defenses. Adaptive defenses or specific include the functions of two things: the B cells and the T cells, right? The B lymphocytes and the T lymphocytes. So essentially, anything that isn't a B lymphocyte, T lymphocyte is part of innate defenses or non-specific. B cells, because they make antibodies and they get released into plasma, is called your humoral immunity, because in Latin, humors mean the fluids of your body. So B cells make antibodies and they go into the fluids of the body. Cellular immunity describes the activity of your T lymphocytes. T lymphocytes will make actual physical contact with the virus to then try to kill it, by releasing its granules on it and let it, pierce the surface of that virus. Okay, so innate immunity. We have a first line of defense. We have a second line of defense. Right? The first line of defense is the physical and chemical surface barriers that prevent entry of a pathogen into your body. Physical defenses are the external barrier created through the epidermis, the stratified squamous epithelium, of your skin. Chemical defenses are things that we can secrete onto the surface of the skin in the case of sweat and oil, right down here. And along the pathway, where you have essentially it's stratified squamous tissue that kind of folds and forms layers and the lining of your nasal parts of your nasal cavity, of your, the oral mucosa, your mouth and your pharynx, where food and things can go, and where your skin from the surface of your body, first infolds and kind of lines like where your nostrils and things are, you have a lot of stratified squamous epithelium. All right, so we're going to be secreting a lot of oil and a lot of sweat there, which then, has certain antimicrobial components to it. As we travel farther in the respiratory system, like up in the nasal cavities, and then where only air travels, like in the larynx and the trachea, and the bronchi, we're going to secrete a lot of mucus there. And that mucus on top of the ciliated cells there is another physical barrier. It helps trap different pathogens and pollen as you breathe in and out. And so it prevent it from entering into your body. Lacrimal, tear production, in your lacrimal glands that bathes the surface of your eye, is another physical barrier as well. So on the surface of our body, we have a lot of physical and chemical defenses to keep something from getting inside. Your second line of defense are some of the reactions once that organism has actually gotten into your body. So the second line of defense are your internal defenses that come into play after the pathogen penetrates the skin and the mucous membrane barriers. It includes antimicrobial substances and those phagocytes. Now, these are still non-specific. The only things that are going to be specific are T lymphocytes and B lymphocytes. Okay, so of the antimicrobial substances, they inhibit microbial growth. Interferon interferes with viral replication. The complement system are plasma proteins that enhance the adaptive immune response. So they're going to help send chemical signals that will actually stimulate those T lymphocytes, B lymphocytes in a way. Transferrin, right? Trans means to transport, and ferrin means iron. Transferrin are molecules that bind to iron that's in your plasma, and it interacts with it and grabs it and transports it essentially out of the blood. Because bacteria like to use iron also. So by binding to these iron molecules, it inhibits bacterial growth by reducing the available amount of iron in the environment where this bacteria is located. So essentially, you're kind of starving them out, starving out your bacteria. All right, so you can stop viral replication with interferon. Know that. We can interfere with bacterial growth with transferrin. Right? Bacteria, transferrin, virus, interferon. Ding, ding, ding. Complement system, no, it's there. I don't really ask too many questions about it, but the C5 proteins in this cascade of reactions, is really active in your immune system. But then we have phagocytosis. That's back to neutrophil and macrophages. They're alerted by chemotaxis. We said that, these cells and the white blood cells follow these chemical scents. And that's where, part of, where when other cells are responding to the presence of different bacteria and things, they're going to release these chemicals into the blood, and the neutrophils and macrophages are going to follow that chemical scent. Natural killer cells are a small population of lymphocytes defending against viruses and cancer cells. They secrete cytolytic substances like a T lymphocyte will, called perforins, that come in contact with the surface of a cell, and destroy the infected cell. I'm thinking of something, and I just can't remember where. I think it was on NPR Science Friday or something, and they were talking about some natural killer cells, and I think it was there was someone they were interviewing, and they're saying these natural killer cells are really important in our existence, in that probably about every 6 months these natural killer cells come across cancer cells that are found in your body. And because there are only a few, a manageable number, at a time, every like 6 months, let's say, is that these natural killer cells are really able to destroy those cancer cells before they really have a time to proliferate, to increase in replication and so forth. And that, it's probably happening about every 6 months or so. Why do cancers progress even further? Because these cancer cells have different ways of adapting to the environment, hiding from the natural killer cells. The more potent they are, it's kind of like different viruses can make you really, really sick, and some of them not too much, like cold viruses. So in any event, these natural killer cells, do a very good job with, killing off some of the cancer cells as they form, but that it's as frequent as maybe even every 6 months or so that you actually do develop these cancer cells. This shows you how a macrophage can capture, these different bacteria. So isn't that cool? They have these extensions and they kind of grab onto them. You're going to endocytotically take these, or phagocytically, take these different molecules, these different bacteria, into the macrophage. You engulf it, you create that phagosome, and then the lysosomes, which contain the digestive enzymes in this cell, are going to merge with that phagosome and squirt out the digestive enzymes onto those bacteria, and that'll kill them. Okay. When we're, and this is describing that activity. Here we have a phagocyte adheres to the pathogens or debris. So when there is an organism outside of one of these cells and we want to phagocytize it, it's going to pinch inward like such and form a vesicle around those bacteria or those particular rods, and we call that a phagosome. The lysosome, or lysosomes that are available in the cell, are going to merge with that phagosome, and you see how you kind of unzip those, those essentially they're phospholipid bilayers, that are creating the vesicular wall here, and you unzip that, and you're able to eject the granules, the digestive granules contained in that lysosome or stored there, and it interacts with that particular bacteria. See, so it says lysosomal enzymes digest the particles, leaving a residual body, and then we exocytotically spit it back out again. Okay, your second line of defense, still nonspecific, is inflammation. The nonspecific tissue response to injury to aid in tissue repair. Signs and symptoms include redness, pain, heat, and swelling. Those are all part of inflammation. redness, pain, heat, and swelling, regardless of whether it's, you cut your finger and you have some bacteria entering in there, you have blood vessel injury, or if you dropped a brick on your foot, you're going to get redness, pain, heat, and swelling because it's non-specific. Right. And then there's fever. It's a systemic response to invading microbes. Increase in temperature increases the reaction rate to aid in repair. It also inhibits microbe growth. So increasing temperature helps, stimulate some of these other non-specific activities, like how interferon and transferrin are going to work, things like that. Remember that the hotter it is, because, we find proteins in bacteria and viruses also, remember, high temperature and low pH denatures proteins. So, when we elevate our temperature like that, because something's in your body and you're not exactly sure what, and we haven't gotten this, the specific system really kick-started, is that if I increase my body temperature, then I create a very inhospitable environment for the proteins of whatever invading organism it is. So you start to at least kill it off, maybe while you're waiting to make the specific antibodies and things. Okay, adaptive immunity or specific immunity is the ability of the body to recognize specific invaders with those specific antigens on its cell surface from your own body cells. An antigen, remember, is a substance that stimulates or can stimulate an antibody response or some kind of immune response in the body. If the antigen is to a foreign invader, we need to get rid of it. If it's that antigen is one of our MHC complexes, we're going to leave it alone because that's a self cell. Adaptive immunity also demonstrates memory. The second exposure creates a more rapid and stronger response. And it involves T lymphocytes and B lymphocytes. Cellular cell-mediated immunity uses the T lymphocytes or the T cell response, and humoral immunity because antibodies, made by the B lymphocytes are technically plasma cells. That's a B lymphocyte that's now a specific factory to make a particular antibody. That functions in the humoral side because we use the fluids of the body to circulate all those antibodies. All right, so cellular cell-mediated immunity is directed against antigens inside a cell, technically. Viruses, transparent to tissues in some cancers. It involves T cells, specifically helper cells and cytotoxic cells. Helper cells help initiate, this entire specific process. We'll come back to that in just a second. Humoral or antibody-mediated immunity is directed against the antigens, because you're going to bind like lock and key to an antigen outside the cell, and provided by antibodies present in body fluids. It involves the B lymphocytes of B cells. Right, so T cells and the cellular immune response. So types of specialized T cells include, we have helper T cells, cytotoxic cells, memory cells, and it just kind of changes every now and then whether they consider this other category of T lymphocytes at the fourth cell or not. The last Myriad, was published. I believe they went back to a 3-cell categorization, of the T lymphocytes. So of the T lymphocytes, we can have a helper T cell, and helper means it's going to help initiate and sensitize, so to speak, the cytotoxic cells here, as well as the B lymphocytes. So it's a required step in initiating your immune response because they're going to help out in this. Okay, helper T cells, sometimes refer, the T4 cells, have what's called a CD4 protein. That's why we call them a T4 cell. Type of protein on the surface. They activate the T8 lymphocyte, the T cells, which are the cytotoxic cells below, and your B lymphocytes or B cells. All right, so helper cells, activate the cytotoxic lymphocytes and the B lymphocytes. No immune response is going to occur without the help of these helper T cells. They are the target of HIV. So acquired immune deficiency, you are making, the organism is targeting the helper T cells. And if you need the helper T cells to initiate your specific antibodies and granules to a particular organism, then that organism is going to be given the capability to replicate over and over and make you very, very ill. Now, it might not affect all your helper T cells because as AIDS or HIV really progresses from initial stages to severe stages, is that more and more helper T cells become affected, and therefore more and more CD8s or cytotoxic cells become affected as well. So you look at what's called, the CD4, CD8 ratios, as HIV progresses, to give you an idea of how progressed it actually is, given the ratio of CD4 to CD8s. There are normal ranges for that. But anyways, so helper T cells are needed to initiate your immune response. All right, then we have our cytotoxic T cells. Those are the ones I said that have to make physical contact to do its job to kill a particular virus, named for the CD8 surface protein on it. Release a variety of granules that enter the target cell membranes, causing it to rupture. Right, so it's going to have to actually physically, one cell to another, make contact, kind of like the infantry division in an army. It requires interleukin, particular chemical, from helper T cells, to work. Memory T cells essentially are what I call leftover army cells. Of these cells, that don't get used, especially the cytotoxic cells that contain the specific granules to kill a particular cell with that particular antigen on the cell surface, those that don't get used up to kill the particular virus, let's say, and that are left over, become the memory cells. They are activated quickly upon re-exposure to that same antigen, so that now we don't have our antigen into the granular, the T cell number of cells, and the antibodies when it comes to the B lymphocytes. Those numbers will increase, very, very quickly, in like a 72-hour period. When we have these memory cells, they can make these specific granules and antibodies much, 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 much faster. And I'm going to show you on a chart like the difference between that. Okay, so T cells and cellular immune responses. So, there's some rules, so to speak. A lymphocyte must be activated before it can respond to an antigen. You don't want an antigen, a lymphocyte, either one, to accidentally identify the antigen on one of your own body cells as being a foreign invader. Because once this process starts, the memory cells will exist there, and you will always, try to kill off these normal cells of your body. That's what an autoimmune disorder is. All right. So to help prevent that, we should have these two signals before we start this whole process of making antibodies and whatnot. So T cell activation, whether it's a T cell or a B cell, requires two signals. First, you need an antigen presenting cell, and that may include macrophages, dendritic cells. Remember, those are found in the epidermis, one of the locations, and B lymphocytes that are memory, it really should say like really memory cell B lymphocytes. Macrophages are really, how macrophages are such, the monocytes, they're the great big white blood cells capable of phagocytosis. When they phagocytize something, they take, because you created that phagosome and now you've broken it down with the lysosomes, is that it's going to take the antigen that was on that foreign cell, and it's going to put it on the surface of its own little macrophage. That becomes another type of MHC complex. Right? That's your MHC2 complex. So that macrophage is going to have an MHC1 really and an MHC complex, 2 complex on it. One of those complexes identifies as being supposed to be there and part of my body normally. And the other complex is a copy of what the virus is. So now that is an antigen presenting cell. It presents that antigen, to the B lymphocytes and the T lymphocytes. Right? So the macrophage, becomes an antigen presenting cell, or APC, because it phagocytes, that's how it obtained the antigen from the virus that it just ingested. Leaves the antigen fragment on its surface for T cell recognition, called a marker. T cells are activated by the presence of this marker. It's, again, simply just another type of MHC complex. So this requires major histocompatibility, or human leukocyte antigens, HLAs, are antigens we use to recognize self-cells. So this is the difference between what we call like those MHC1 complexes, being able to identify our self-cells, versus an MHC complex 2, which is one of these fragments from a foreign invader on the self-cell. Okay. In addition to having some kind of antigen-presenting cell, so on the most part, it's going to be macrophages, but it could be, a B cell, and it could be a dendritic cell. They're going to do somewhat the same thing. They're going to bring the antigen, to the helper T cell, and then the helper T cell is going to stimulate the T's and the B's. Okay, but we also need a co-stimulator. We need two signals: a chemical signal called a cytokine, produced by an antigen-presenting cell surface that has been damaged or invaded by pathogens such as interleukin-2. So another type of a chemical is called interleukin-2. And between the antigen-presenting cell and this other co-stimulator chemical called interleukin-2, now we know, okay, we for sure want to start the process of making specific T lymphocytes and B lymphocytes directed at that particular antigen. Humoral immunity involves B cells that remain in lymphatic tissues, lymph nodes, spleen, and lymph nodules. Antibodies, that are going to be made in these B cells, which are now called plasma cells, and they get released by massive numbers, into plasma. All right, so now they're going to be free floating, kind of like how your anti-A's and anti-B's would be floating in blood. All right, they're going to be floating around in the plasma. So the limitation of the humoral immunity is that those antibodies will only go where those fluids can, be transported through the cardiovascular system. Right. Produces two types of cells. The plasma cells are the ones making and secreting the specific antibodies. And the memory B cells, like a memory T lymphocyte, the B lymphocytes kind of left over after eradicating the virus. Upon later exposure, these cells can quickly differentiate back into plasma cells, which is an active factory making that specific antibody again. All right, that takes us to an immunity video. I don't know if I run it, if it's going to stay in my share. What you can do is you can watch this on your own. When you download your PowerPoint and you put it in slideshow presentation like this, it will, run the video. If I don't forget, I will quickly come back before I'm done, because we still have, a few more slides to go, and I would try to stop talking quite so much. And then I'll try to remember to come back to it. And if I can run it and change the share, then great, and if not, then we'll just end the video. Here's a K2 quiz. We're clearly not going to be able to have participants in this. So in a live class, face-to-face class, I would open up this quiz and you can play on your phone, but we're recording this presentation so you can view it later. We have no, this isn't live online. So therefore I have no barking dogs in the background like I did with some of my former videos, when we first started the pandemic. So I'm going to continue on, and I will do the best I can and to come back, and so we can quick watch that video, and hopefully it will be shared with you, and then I will, end this presentation and put it up on the course page. Okay, so antibodies, we need to cover some other aspects of different responses and how we're going to respond to different infectious agents, in our body. Well, the antibodies, and this is good to keep in mind and look at these different classes and the shapes of them, where they come from, because you're going to use that in microbiology as well. Antibodies, their actions are neutralization. They can block receptor sites on viruses, so they can't bind to the host. So what antibodies are going to do is they're going to bind to the particular antigen on a virus, and this helps to neutralize them so that virus can't get into your other body cells, because viruses enter your your body cells and use all those organelles, like your own nucleus, your ribosomes, and your ability to make proteins, and they live off your normal, body cells, and they take all the nutrients there. Those viruses replicate over and over again, and then they kill the normal body cell. And now you started with one virus, and now you're liberating like 100 viruses into plasma. So it helps neutralize them so they can't invade our host cell. Agglutination precipitation, when antibodies bind to many of the different antigens that are present on the surface of a virus, then it causes agglutination. Like when we blood type blood by adding, let's say, anti-A or A antibodies to a droplet of blood that we're not sure, we don't know what the blood type is. And so if A antibodies are put into contact with blood and there are A antigens on the cell surface, it binds to it, right? And that made that clumping effect, that was called agglutination. And if you have enough agglutination, it will precipitate out of this solution and fall to the bottom, right? So it helps bind to them all. And then it stimulates the complement activation, that series of different, a cascade of reactions that produces those complement proteins. Those are plasma proteins that help destroy antigens by causing phagocytosis, cytolysis, meaning just breaking the cell entirely, and inflammation. That's where that C5 protein comes in. Okay, so how do antibodies work? Right, so these B lymphocytes, once they're sensitized to produce an antibody to a specific antigen, they turn into what's called like an antibody factory, and that's what we call kind of a plasma cell. They make tons of antibodies there, and then it kind of exocytotically releases them so that they're floating around in plasma like your anti-A's or anti-B's would do. So here in green, see those green Y's? That's what's considered a monomer, a single unit of one of these antibodies. And they're going to float around in plasma. And what they're doing is we're waiting for those to collide with a complementary antigen on the surface of a particular pathogen. So they're the pathogens in pink with the red little triangles on the surface. And when the green antibody comes in contact with them, the binding site there at the tips of those Y's or antibodies lock and key bind to the shape of the antigen on that pathogen. And thus, the more antibodies we have, the more we can kind of cross bridge, like over here on the right-hand side to the bottom here, and you can cross bridge and form these larger and larger clusters that not only inactivate them and neutralize them because now they can't get into another cell, but, it helps us to identify them, to hold them in place until a T lymphocyte might come along and, spit out its killer granules on them. Okay. Now, the other fate, is that over to the left-hand side here, it's showing you that they could be phagocytized by maybe a macrophage. Okay, characteristics of major immunoglobulins. So we have 5 classes of antibodies, so what's called immunoglobulins. And the mnemonic I always used was GMADE, G, M's, ADEs, because G's are the most frequently occurring, and then M's and A's and D's, and then E's. But your textbook kind of approaches it this way. All right, so IgMs, like macrophages or monocytes, they're the big mothers, right? IgMs, it is the first antibody to appear in response to an initial exposure to food or bacteria. So when it comes to, some of these immune responses, IgMs are going to elevate first, and, to bacteria and to different exposure to foods that could be that you're allergic to. Examples of these larger ones are anti-A and anti-B antibodies that you automatically make in your liver. But these are going to be made in plasma cells. So IgMs is going to be an example of a pentamer shape. I'll show you in just a minute. They're huge. They have 5 different of those Y monomers, attached to it. Okay, then we have IgGs. It's the only antibody capable of crossing the placenta to impart passive immunity to a fetus. Good to know. It's very small, very effective against bacteria, viruses, and toxins, and that it constitutes generally the highest concentration or quantity of immunoglobulins that are circulating. Just like white blood cells, you had the most common, and then the second most common, neutrophils and lymphocytes, the least frequently occurring, but the basophils. Well, it's the same when it comes to these antibodies. However, when you get exposed to certain things, just like when, your white blood cell lines will increase when you get exposed, maybe more specifically to a parasite versus a bacteria versus a virus, is that these will elevate when you get exposed to certain things as well. All right, so the IgGs, are the only ones capable of crossing the placenta. They occur most frequently, and they're the highest population. It goes Gs to Ms, then the As, the Ds, and the Es. The IgAs are found in fluids of the body, so the way IgA antibodies will be imparted or transported is through saliva, tears, and breast milk. So again, breast milk after birth, another way of imparting some passive immunity to, the infant. IgDs are the ones found on the surfaces of most B lymphocytes or B cells, and they help function as a B cell receptor. And then IgEs, kind of like eosinophils, play an important role in allergic reactions. Example would be hay fever. That's a environmental allergy, to a particular, to pollen. Okay, antibody components. When it comes to an antibody structurally, we have what's called the basic antibody structure. Overall, it's a T or a Y shaped, and most of your diagrams in your books are going to have a Y shape to them. It's a Y-shaped antibody monomer, which means a single unit and consists of 4 looping polypeptide chains linked by a disulfide bond. The 4 chains consist of, out of the 4, 2 heavy chains and 2 light chains. The 2 heavy chains, with a hinge region at the middle, and then we have 2 light chains, and I'll show you where they are on the diagram in just a second. Then you have your variable region. Those are at the tips of the Y, so to speak. At one end of each arm, combined to form 2 identical antigen binding sites. Okay, so here we have that kind of Y shape. The heavy chains are to the center in like the light bluish and dark bluish areas. And then the light chains, are there in the purple. All right? The arms of the Y, where you have the double strand there of blue and purple, all right, at the tips of those, that is the specific antigen binding site. That is called a variable region, because these regions in the light chains and the heavy chains thereafter, and you see the hinge point there, and then your stem region, it's there at that variable antigen binding site. Well, you will have the specific keyhole to have the key fit into. Okay, so we want this to have a complementary shape to the antigen so that we have a lock and key mechanism here. Right? And that variable end at the tips of the Y's there, is going to change. Right? This is an example of, so over here on the left-hand side, here we have one of the monomers. It kind of looks like a Y, right? We said the most of them are going to be Y-shaped. Here at the tips in yellow is the specific binding site. This is the variable region. It will vary depending on the shape of the antigen, which is right up top there. They're giving you an example of a purple one, a red one, a green, what is that, tan, blue, and gold. The different shapes of the antigen will determine the shape of the binding site. So it's a lock and key. And that's what makes them so specifically, functional like this. Because the only thing that will bind to those binding sites on that antibody is the specific antigen of the particular organism you're trying to kill. All right, so over to the right-hand side shows you an example of that Y, the binding site over at the ends here. All right, examples of that single unit monomer are IgDs, IgEs, and IgGs, right? Ds, Es, and Gs are GEDs. Well, that's a way to remember it, a GED. You can have a 2-unit, 2 monomer antibody. That's called being the dimer shape because there's two, and that's the shape of IgAs only. And then the pentamer structure, 5 of these monomers, right? That's the big mother. I call that the IgM. Where on each arm, of these monomers, you can have out of these 5, components, because the pentamer means 5, you have 2 binding sites on each. That's the ability to bind to 10 10 antigens. So these pentamers do a really good job once they get in an area where all these viruses and things are located, is boom, 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 being able to bind to maybe 10 of them. Okay, so homeostatic imbalances, parasitic infections by worms such as Ascaris and Schistosoma require different immune attack strategies. Some worms, are too big for regular what's called plant attack, precipitation, lysis, agglutination, or neutralization. So IgE antibodies can still play a critical role in the worm's destruction by binding to the surface of that worm. All these IgE antibodies are going to start attaching to these antigens because, they have more than one. It's not like they have one antigen on their cell surface. They have hundreds, if not thousands, depending on the size. Right? And they mark it for destruction by those eosinophils. Remember, eosinophils elevate, with almost uniquely with parasitic infections. Okay, so the eosinophils then bind to the exposed stems of the IgEs, which triggers eosinophils to release their toxic contents onto the prey, lysing it from inside out. Isn't that cool? Yeah, so that's an example. Okay, immunological memory. So when it comes to, and this is sort of the basis of why vaccines can work so well without getting into politically all that stuff, is that, when you get exposed to something new that you've never seen before, that is your primary response, your initial exposure. And when that happens, the binding of an antigen to a cell causes your T and B cells to become activated and what's called immunocompetent, matured. So they start multiplying rapidly, differentiate, and start making either the antibodies by the B cells or the granules by the T lymphocytes for a specific, organism, for that specific antigen. Cytotoxic Ts, the helper Ts, and the plasma cells, which are the activated B lymphocytes, eliminate your initial infection. Plasma cells make the antibodies, and the cytotoxic cells find those and they squirt their, granules on them. They kill, that particular virus, let's say. The memory T and B cells, that which is left over from, winning this battle against this virus, will be used in the secondary response. They're going to live on, right? Those memory T cells are going to live on, right? The primary response, this book, will indicate it's about 21 days before you really peak at making your antibodies. And that, I think it's a little bit longer than it probably, I'd say that more than likely about 2 weeks to maybe 3 weeks is when you'll peak in the amount of antibodies you're making. But upon secondary exposure, it takes a fraction of the time for you to max out, not only for you to max out the antibodies you're going to make about 4 times as many antibodies as you did from the primary response. And you're going to do it, in a quarter of the time, if not less. So your secondary response, a subsequent exposure, those memory T's and B's are in place, boom, and they quickly activate to fight against the antigen. Faster, more prolonged, more effective response than the primary response. So here, and this is the one that, I don't really care too much. I need to check their newer edition and see if they've changed this, but the blue, right, line here, it's up at the top here, says this is your primary immune response to an antigen, A occurs after a delay. All right, so down on the bottom, it's saying, look, let's start with day 0. On the horizontal, we have day 0, 1 week, 2 weeks, 3 weeks, 28 days, 35, and so forth. And on the vertical y-axis, this is the antibody titer, the antibody concentration, the number of antibodies you're making, or you can make. So they're saying, okay, let's say in blue, we have our first exposure and the arrow is pointing to day 0. Right? right? It's going to take, here at about day 5 to 6 is when you start making these antibodies, right? And so antibodies to A, here we're, maxing out here. And then slowly those antibodies decline as you kind of use them up, in trying to kill the virus. Right? And so it slowly declines and wanes till you get to about day 28. But then we have a second exposure. So let's say we get exposed to that same virus again. So they're saying second exposure to antigen A, first exposure to antigen B. Right. And you get exposure to the same virus versus now a new virus. Well, the response to the virus A, which you've already been exposed to, look how quickly we make those antibodies. We do it in not only a fraction of the time, but we make about 4 times as many. So here it says up at the top, secondary immune response to antigen A is faster and larger. Primary immune response to antigen B is similar to that of antigen A. It's going to take a while. It takes a while, not a long time, but it takes a while before you really, figure out the shape of the antibodies. You clone those plasma cells so that you have, instead of one factory making antibodies, you now have, 100 making antibodies. Right, so it takes some time. That's the whole point of vaccinating, is that, if you can introduce, a dead virus, and sometimes it's a live virus, and now we use messenger RNA, which bypasses this and makes your response even faster, is that if you give a small quantity that's of something that's not going to get you sick, it stimulates this whole process and you stay asymptomatic. Right. And so you develop these memory cells. So if you do come across that organism, and sometimes it doesn't have to be the exact same organism, it could be one that has a very, very similar antigen on the cell surface where maybe the same antibodies you have, might be able to bind to that. Let's say antibody, the B antigen here is very, very, very similar to A. Some of those A antibodies may actually be able to bind to that B antigen. We'll still make them specific for that B, as we go along, but sometimes it can be kind of do double duty. Okay, acquire adaptive immunity. So when it comes to immunity, we have 2 different categories, and within each 2 more. Active immunity versus passive immunity. Active immunity means my body is making antibodies. All right. I'm not getting antibodies someplace else, like through mother's milk or across the placenta or in an injection form, because you can, sort of artificially, so to speak, create antibodies for a particular virus or antigen, and just like transfusing blood by putting just the red blood cells into a patient, you can just get specific antibodies for a particular antigen and inject it, right? But active immunity means my body's going to make those antibodies. So naturally acquired active means, like I've come across, which I did, just in the last few weeks, is come across something, I think it was a cold virus, a rhinovirus, and I got exposed to enough of that virus to make me ill. That virus starts affecting my body cells, and I now have to make the antibodies to it and slowly kill it off. But in the process, I will hopefully I will have these memory cells. So the way I developed my immunity for the next, not only helped me in the first exposure, but it's going to help me in the second exposure, is that I actually got a little sick somehow, and then I made my antibodies. Right. Artificially acquired active, artificially stimulating my body to make antibodies is how these vaccines work. Stimulates the primary response. You don't get sick from it. They're generally dead viruses. Occasionally, a small quantity of a live would do. And as I said, in the case of COVID, we used messenger RNA, which is fantastic because your response and the way you make the antibodies, or the speed in which you respond is going to be faster than the old-fashioned way, of either having to come across it. Of course, that would be horrible, or having a vaccination from the dead virus of COVID-19. So the messenger RNA circumvents, that requirement and just gives your cells essentially the recipe to make the antibodies without ever having that live or dead virus enter your body. So I think that's way, way, way cool. Okay, then we have passive immunity. Antibodies are acquired from another source. We can have naturally acquired passive. Examples of that are mother's antibodies, either the IgGs that can cross the placenta or the IgAs in breast milk to an infant. Artificially acquired passive is the method of injecting antibodies. It's an immediate protection. It's more short-lived. You might have to get multiple injections. All right, so you should know the difference between these types and what it means to be naturally active, artificially active, naturally passive, artificially passive. Okay, and so here, as part of your humoral immunity, you have your active versus passive methods, as just described. Natural active, infection comes in contact with the old-fashioned way of getting sick and making antibodies. Active artificial is like a vaccine that gets injected, right? Natural passive would be getting antibodies maybe passed from the mom, either across the placenta or in breast milk, and artificially passive injection of what's called exogenous antibodies that, from another source that get injected into you. Okay, FYI, lymph locations of lymph nodes. I think this is really handy to have, especially those of you that are, probably going to go into nursing or into PA school. These are the chains, common chains of lymph nodes, for that you might want to and have to examine upon physical examination. Those in the abdominal cavity and the pelvic cavity and the thoracic cavities deep away from the surface of the body are not going to be palpable. But if they become infected, they may be seen possibly on X-ray and or nuclear medicine scans. So we have a cervical region, the axillary region, which is the armpit, supratrochlear, which is the elbow, the anterior elbow, the inguinal region, the bend of the hip, the pelvic cavity, abdominal cavity, and the thoracic cavity. When it comes to around the head and neck, and these are palpable, especially if they become, if they become swollen or affected and active because they are capturing, different virus or bacteria, and or they might be the target of metastasis, unfortunately, in the case of cancer. We have what's called the preauriculars in front of the ear, postauriculars. We have the occipitals or suboccipitals. We had, this is where sometimes they throw these together, the parotids and the tonsillars. We definitely have the submandibulars and submentals below, around the angle of the jaw, underneath there. That would be your submandibular, and then the submentals, the mental region is the anterior aspect of the chin, just underneath there. We have superficial cervicals, deep cervicals, posterior cervicals, and the supraclaviculars. Those are the ones I said, those are fairly easy to palpate if they're involved for sure. Again, upon reaction or finding some kind of finding there, it always requires some kind of follow-up as to the reason why you find these either swollen or larger, harder, and painful. Sometimes they're painful, sometimes they're not. Okay, then we're going to talk about hypersensitivity reactions. These are not part of FYI. The FYI was really these locations and things, but I think they're, quite helpful. All right, hypersensitivity or allergic reactions. We have 4 different types. We have type 1s, type 2s, type 3s, and type 4s. Type 1s occur in minutes after contact with an allergen, when it's an allergy, right? Symptoms include hives. If you've ever had hives, you know what I mean. I have very strong reactions to hay fever. It really causes this type 1 hypersensitivity, very severe. Asthma, eczema, gastric disturbance, and anaphylactic shock are all fall into type 1 hypersensitivity reactions. They're rather severe, and they occur very, very rapidly within minutes after you come in contact with something. Okay, type 2s take a little bit longer, sometimes 1 to 3 hours to develop. An example is a transfusion reaction. Now, I will tell you, it doesn't take hours for you to see a transfusion reaction, especially if you have mismatched blood, and you have mismatched, or someone has somehow given, let's say, where you have not matched up Rh positive and Rh negative reactions in someone that is sensitized. When you give B blood to an A blood type person, that A blood type person has all these B antibodies. And if you put the red blood cells that have B antigens on the cell surface, into that patient's body, then their antibodies are going to bind to those A antigens on those red blood cells, and you're going to have hemolysis. You're going to start killing off those red blood cells very rapidly, and that's an example of a transfusion reaction. You're going to spike a temperature, they're going to hit your spleen and ultimately your kidneys, and you're going to see a severe response. Okay, type 3 reactions, they may take about 1 to 3 hours as well. These may, but the severity can vary. So immune complex reactions, usually using the IgGs, which are those little ones, and the IgMs, those are the big mothers. It may take an hour or a couple hours to develop. Antibody complexes cannot be cleared from the body. And then you have damage to body tissues. So an example of that is rheumatoid arthritis. So, these responses, it doesn't mean they massively happen all at once, but they may be chronically occurring, because rheumatoid arthritis, left on its own, is not going to go away. You're going to create these, you're making antibodies, G's or M's, to bind to these cells, and that they form the antigen antibody complexes, kind of like what we've seen, when we're blood testing, and they will often precipitate into joint surfaces, in the joints where they're being formed. And, that's forms rheumatoid arthritis. Or in the case of rheumatoid, we're going to have deposition. Those antigen antibodies aren't going to be cleared, and they deposit into the joints, and so we call that whole presentation, so to speak, and process, that's rheumatoid arthritis. Type 4 hypersensitivity reactions is kind of like a delayed reaction allergy kind of response. And an example of that is contact dermatitis. So an example of what contact dermatitis means, it means that this substance has to come in contact with your skin. It might actually get into your eyes and into your respiratory system, because some of these oils kind of aerate, become particulate in the air. When you breathe them in, then it's going to come in contact with your pharynx and along your respiratory tree, and then you may have this type 4 reaction as well. But an example is, let's say, poison ivy, for those of you who know what that is, or maybe if you're allergic to certain soaps for, doing your laundry soaps, and sometimes that soap doesn't really get washed off well, some of the scents, and there's a residue from those soaps. And when you put your clothing on, it interacts with your skin, and now you're getting, an allergic response. It produces eruptions and inflammation of the skin, and often takes about 48 hours to occur. All right, so it takes, not a long period, well, not a long period of time, but longer than the type 1, 2, and 3, and then you see, an indication of this hypersensitivity reaction, on the surface of the body in these pustules or eruptions and redness, and sometimes, itchiness of the skin. Autoimmunity is when the immune system fails to distinguish your self cells from your non-self cells and produces antibodies called autoantibodies and cytotoxic T cells to attack the body's tissues and organs. So you shouldn't make antibodies to antigens normally found on your cells. And this is a list of different types of autoimmune disorders. Some you may be familiar with, and some you may not be. But in any event, glomerulonephritis, in his example, Graves' disease, we'll talk about that a little bit more, and type 1 diabetes mellitus when we get to the endocrine system. We've already talked a little bit about hemolytic anemia in the blood chapter. Myasthenia gravis is a new one where we make antibodies to the receptors for acetylcholine on the sarcolemma of skeletal muscle. Pernicious anemia, that was again another one that we somewhat mentioned in the blood chapter. rheumatic fever, rheumatoid arthritis, we just mentioned, systemic lupus, and ulcerative colitis. So, I'm not going to ask you about these. I mean, where they come up in previous chapters, they really would be more relevant to those chapters, and I'll probably leave it at that. I think I would know those that I referred to, in like hemolytic anemia, pernicious anemia, in the blood chapter. But especially if you know anybody with these diagnoses, now you can kind of familiarize yourself, with what makes this particular autoimmune disorder different from other autoimmune disorders. Okay, so that's the end of the presentation. I'm going to quickly go back and see if that video will run. I'm going to invoke it, and then I'm going to change my share here. So I'm going to stop my share, and then I'm going to, let's see if I can get this to run, and let me actually change my share here. Okay, let's see if this will work. Let me turn up my volume, and let's see if this will go. Woohoo!
This patient's cold is caused by rhinoviruses, shown here in red, that entered her nasal passages and escaped the first-line innate defenses, such as mucus and cilia. Some of these rhinoviruses entered the respiratory epithelial cells, where they replicated, releasing more rhinoviruses into the patient's nasal passages and tissue fluids. In the tissue fluids, cells of the second line of innate defense, such as this dendritic cell, carry out surveillance for anything that appears foreign. Dendritic cells engulf the rhinovirus within an endosome. The endosome fuses with a lysosome, whose enzymes digest the rhinovirus. The resulting rhinovirus fragments are called epitopes. The dendritic cell then attaches the rhinovirus epitopes to presentation molecules called MHC2. This complex folds outward so that it sits on the outside of the dendritic cell membrane. The dendritic cell then enters a lymphatic vessel and travels to a nearby lymph node. Here, the dendritic cell, part of the innate defenses, encounters a helper T cell, which is part of the adaptive defenses. The T cell receptor, or TCR, of this helper T cell recognizes the rhinovirus epitope, while the CD4 glycoprotein associated with the TCR recognizes the MHC2. The dendritic cell then stimulates activation of this helper T cell. Adaptive defenses help to control the current infection, but also provide immunity against future infections by the same rhinovirus. They are carried out by two types of lymphocytes, B cells and T cells. Immature B cells conduct surveillance in the lymph nodes. Using B cell receptors,
I'm going to stop just for a second. I want to mention two things, and then we'll keep going. I believe what they referred to, as the endosome is the same as a phagosome, which is the term that your textbook uses. The rest of that is fine. And then when they just showed you how the antigen presenting cell, went to the helper T cell, how it recognizes the self cell, the MHC complex and whatnot, in addition to having that helper cell, it doesn't mention the chemical stimulus it needs as well. You need these two conformations, the helper T cell and the particular, like the interleukin-2, stimulate this process. So they're not exactly saying exactly what's said in your book, but I think it's giving you a good pictorial, or a visual image of what's happening. I think it's pretty darn cool. If there's something else that's kind of off kilter a little bit with your textbook, I'll stop it and mention it. And otherwise, I think this is really cool.
Or BCR, to recognize only one specific epitope. The BCR on the left does not recognize the rhinovirus, but the BCR on the right does. And this B cell is selected for activation in a step called clonal selection. The selected B cell takes in the rhinovirus, degrades it, and presents the rhinovirus epitope with its MHC2.
And I'll just gotta stop it there again. So what they're talking about, the epitope, is the antigen on the cell surface, right? The same thing.
Our mature helper T cell recognizes the rhinovirus epitope with its TCR and the MHC2 with its CD4. Then the helper T cell secretes chemical messengers known as cytokines that activate the B cell. The B cell proliferates and differentiates in a process called clonal expansion, resulting in two types of cell. Memory B cells carry the memory of this pathogen for many years. Plasma cells secrete antibodies that conduct surveillance for more rhinoviruses, grouping them together so they are easy targets for phagocytes. Antibodies in the blood and tissue fluids seek out free rhinoviruses. The antibodies neutralize the rhinovirus by binding to its surface molecules, thus preventing attachment to host cells. Antibodies are humoral defenses that target free pathogens. But what about the rhinoviruses that are already inside our patient's cells? Another type of adaptive defense cell, called a cytotoxic T cell, can locate and destroy rhinovirus-infected cells. It provides cell-mediated or cellular immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by a CD8 glycoprotein on the cytotoxic T cell. With the help of cytokines from a nearby helper T cell, the cytotoxic T cell is activated and clonal expansion occurs, resulting in some long-lasting memory cells ready for a subsequent exposure, and many active cytotoxic T cells, or CTLs, that leave the patient's lymph vessel to search for infected cells that present rhinovirus epitope with their MHC1. The epitope is recognized by the TCR of the cytotoxic T cell, and the MHC1 is recognized by the T cell's CD8 glycoprotein. The cytotoxic T cell then secretes perforin molecules, which form channels in the infected cell's membrane. Granzyme, also from the T cell, passes through the perforin channels and activates enzymes that lead to apoptosis, the programmed death of the infected cell. The cytotoxic T cell then moves on to look for another infected cell. Together, the humoral and cellular defenses are the body's third line of defense in fighting a very specific pathogen. The humoral defenses, ultimately in the form of antibodies, conduct surveillance for rhinoviruses that are outside the body cells, in the patient's tissue spaces and bloodstream. The cellular defenses, in the form of cytotoxic T cells, search for rhinovirus-infected cells, inducing apoptosis when they are found. Memory T and B cells help our patient's body to recognize and more rapidly fight the same rhinovirus should it infect her again.
Okay. So, I think that's pretty good. You can see there's a lot of detail to this, and I tried to find a video that would most closely coincide with the book. So there's some minor differences and things there, but you get the concept of how this is so specific, and how one system works the other. One is using the antibodies, B lymphocytes, then the T lymphocytes, using the granules that it produces and it comes in contact with the cell, the methods of identifying whether they're MHC1s or MHC2s, I wouldn't really worry about any of that really too much, but you get the concept of that, and how together we have to have antigen presenting cells and so forth, how, we have to be able to identify our self cells from our non-self cells, and some of the terminology is a little different, but the process, is about the same. So anyways, okay, well, that is the end of the presentation. I hope you enjoyed that and you found the video helpful. It's sometimes hard to find videos that will totally coincide, off the internet or whatever on YouTube, with the content of a textbook, but, I think it gives you a very good visual representation. All right, so that is the end of the Zoom.